So how are enzymes catalysts for reaction? What does that mean? Well, molecules aren't just going to react with one another unless they're provided with some energy, okay? And that energy is called the energy of activation. Now, how enzymes make this easier is they lower the energy because they're able to bring molecules into contact with one another for the reaction, in order for the reaction to happen. So, if if I'm the enzyme, okay, and I need to bring you in the left corner of the room over to your classmate in the right corner of the room. Well, you guys kind of stay in your corners. You might not interact in any way, but I need to bring you two together to make a product, okay? So I, the enzyme, will actually bind with you, walk us over and bind with your other classmate, and make a product. So I lower the energy needed for that to happen because I put you two in physical contact with one another. So if you look at the amount of energy here on the Y, and then the progress of reaction on the X, without an enzyme, which is the blue, there's quite a bit of energy needed for molecules to react. But with an enzyme, aka me, I lower the energy of activation by bringing molecules together.
Enzymes are proteins that speed up chemical reactions in the cell. A special region on the enzyme, called the active site, has a shape that fits with specific substrate molecules. An enzyme works by binding to one or more specific molecules called reactants or substrates. Binding occurs at the active site. The enzyme and substrates form an enzyme-substrate complex. The interactions between the substrates and the enzyme stresses or weakens some of the chemical bonds in the substrates. These stresses encourage a link between the two substrates, leading to the formation of a different molecule. As a result of the chemical interactions within the active site, a new product is formed. The product is released from the active site. The enzyme assumes its original shape and is free to work again. Although this reaction has specifically illustrated the formation of a single product from two substrate molecules, other enzymes catalyze the formation of two products from a single substrate.
So, enzymes actually change their shape to smush substrates together and make one product, or pull substrates apart. So they're actually kind of contorting and changing their shape after the substrates bind. So the active site is the specific part of the enzyme that fits with the substrate. So in the video, these right here, this part right here is the active site of the enzyme, because this is the part of the enzyme that fits with the substrate. And when the substrate binds, the active site changes shape, and that's called an induced fit model. So the enzyme undergoes a slight alteration. Now, in the video, it showed you putting two substrates together to make one product, and that would be synthesis, joining molecules together. But enzymes can also split molecules apart, and that's called degradation. So in these two examples here, we have our substrate binding to the active site of an enzyme. That forms an enzyme-substrate complex. And at the end, we get two products. So this is an example of degradation because you start with one substrate and you get two products. Now here's the other cool thing about enzymes is that they don't get used at all in the reaction. So the enzyme bonded, release the products, and then it can go on and facilitate another reaction. So enzymes don't get used up, they get recycled. That it can go on and bind with more substrates. And then alternatively, you have two substrates binding to the active site of the enzyme, forming the complex. The enzyme changes shape and forces the molecules together to make one product, and the enzyme can go back and facilitate another reaction. So we have degradation here, synthesis here. Now, there are several factors that can change how fast these processes work. The first two factors are by changing the concentration of substrate and enzymes. So, as you can imagine, right, the more enzymes or the more substrate you have, the faster the reaction is going to go. Enzyme activity will also increase with temperature because, right, when you add energy to molecules, they vibrate more, and so that increases the chance of collisions between the enzyme and the substrate. But this will only increase up to a certain point because once it gets too warm, then you can actually break the bonds within the enzyme protein, and that's called denaturing. So hot temperatures can be too hot for the enzyme. It'll actually denature it and destroy it. And this is one of the reasons why having a really hot fever is dangerous for your body, because if your body temperature gets too hot, then you can start to denature the enzymes. Another factor is pH. Most enzymes are optimized for a particular pH, so it's not that, oh, if it's more acidic, it's bad, or if it's more basic, it's bad. It's basically if it's different, if the pH is different from what the enzymes are optimized for, then that will lower enzyme activity. So, I told you that if you change the enzyme or substrate concentration, that will increase the activity of the enzyme. So, in this figure, you have rate of reaction on the Y. Here you have enzyme concentration on the X, so this is saying this is increasing enzyme concentration. And then what's listed in parentheses is really important, because this is saying, okay, we're going to increase enzymes, but we're always going to make sure that the substrates are in ready supply. So if the substrates are in ready supply and enzyme concentration goes up, so will rate of reaction. Okay. Same graph here, except now we have substrate concentration, and now instead of enzyme concentration going up or being in excess, enzyme concentration is constant, which means there's only a limited amount. So as substrate concentration increases, so will the rate, but at a certain point it starts to level off now, because now there just aren't enough enzymes to facilitate the reaction for all of the substrates that are in the system. Okay. Now, in this graph, if I said that substrate concentration was constant, this line would look exactly like this line, because enzymes and substrates are dependent upon one another. So if you limit one of them, that will limit the rate of reaction. Okay. So that was concentration. Temperature, I told you, is really important for the rate of reaction. So in this picture, I have a cold-blooded animal and a warm-blooded animal. So what does that mean? Warm-blooded animals can regulate their body temperature on their own. Cold-blooded animals have to use the environment to regulate their body temperature. But why is that a thing? Like, why is it important? Why is it bad if you get too hot or too cold? Well, that's due to your enzymes. So, rate of reaction on the Y, temperatures on the X. We want to make sure that we keep our bodies at the temperature that yields the highest rate of reaction for enzymes. Because that means that we're being really efficient. Our metabolism is being as fast as possible because our enzymes are working as fast as possible. So our body temperature is what, 98.6? And the temperature of your home is probably 70. So we are always warmer, not always, but usually a lot warmer than our environment. So we're constantly losing heat to the environment. So you might think, well, that's kind of inefficient, right? Like, if we're trying to keep our body at this hot temperature, but we're constantly losing heat, what's the plus? Well, the plus side is that our enzymes and our metabolism works the best at that temperature, and that's why our bodies stay at the temperature that they are. We also talked about pH. and how changes in pH can affect enzyme activity. So looking at this graph, we have rate of reaction on the y, and we have pH on the x. So there's two enzymes here, pepsin and trypsin, and you can see their rate of reaction curves. So which of these enzymes is the one that's found in your stomach? Which of these ones is one found in your stomach? If you said pepsin, you are correct. So pepsin has the highest rate of reaction at a pH of 2, which is the average pH of our stomach. Now, if we put pepsin in our intestines, which is a much higher pH, the rate of reaction of pepsin goes way down. Alternatively, trypsin is the enzyme found in our intestines. If we put trypsin in our stomach, the rate of reaction goes way down. So it's not acidic or basic, it's just outside of the optimized pH range can negatively affect enzyme activity. Okay, the last factor that affects enzyme activity are the presence of cofactors. So you might not necessarily want all of your enzymes on all the time, right? Because at some point you're going to start running out of substrates, and it'll just, or you have plenty of product, you don't need to make any more product, let's turn the enzyme off. And so our body can activate or deactivate enzymes using cofactors. Now, cofactors are non-protein molecules that are mostly made up of vitamins. And what cofactors do is that they change the enzyme shape. And by doing that, that can turn the enzyme either on or off. So some cofactors bind to the active site of an enzyme and change the enzyme shape so that the substrate can fit. Okay? So would this be an example of turning an enzyme on or off? This would be turning an enzyme on. Without the cofactor, the substrate can't fit. But once that cofactor is bonded, now the substrate has a perfect fit.